So, hey guys, welcome back. This lecture is going to be about microbial genetics. We're going to talk a little bit about how DNA and how it works and how DNA actually encodes for things that the cell needs to do. So we're going to delve into what exactly that means. So things we're going to talk about here: features of DNA and RNA, process of DNA replication. We have to make copies of it, right, in order to make new cells. The three processes of horizontal gene transfer in bacteria, which are called conjugation, transduction, and transformation. We'll talk about how each of those work. The mechanisms of expressing a protein, the steps of transcription, which was DNA to RNA, and then translation, which is RNA into protein, and how that all works. We'll talk about inducible and repressible operons. Basically, operon is like a system of genes that kind of depend on products and stuff, as far as whether they're turned on or turned off and everything like that for bacteria. And then we have the effects of different mutations in protein expression. So literally what happens whenever things go wrong, if there's a change in the DNA that shouldn't be there. So understanding what genetics is, genetics really is the term referring to the study of inheritance or heredity that explores transmission of biological properties. In this case, we're talking about like traits, right? Like eye color and stuff like that, or whether you have 5 or 6 fingers on one hand, from the parents to the offspring, and which ones are going to be expressed, right? So how those traits are expressed. The structure and function of the genetic material itself, the DNA and all of that, and how the material changes, if it changes, and why, I guess. You want to ask the bigger picture questions. Anyways, moving on. The genome, we talk about the genome, we're literally talking about all of the genetic material in the organism. We're not just talking about the chromosome, we're talking about everything that could code for anything in there. So most of it is the chromosome, the chromosomal portion, but we can also have plasmids in the case of bacteria, which are tiny little extra pieces of DNA. Plasmids are circular. Just FYI, and that is something you need to be aware of. And that would be an example of part of the genome that we found in non-chromosomal site, or what we would call extra chromosomal. Okay. Then we can also have mitochondria and chloroplasts, like your cells, my cells, we have mitochondria. Plants might have chloroplasts, or other organisms that have these things as well. But just those are the biggies. And those organelles, those particular structures inside of the cell membrane mound, right? They have their own genomes, or chromosomes, really, I should say. That's his chromosomes, believe it or not. So anyway, I don't want to go back and fix it. Genomics is just the part of science that has to do with studying an organism's entire genome, so not just their chromosomes, right? But what other aspects they might have. It's a really big deal with bacteria because those plasmids, man, those plasmids can really cause a lot of trouble. So eukaryotes, they can also carry plasmids, but mostly we're just talking about fungi and protozoa. We're not really talking about the bigger eukaryotes like you and I. And then with viruses, we've got DNA or RNA, but never both, right? Could be single-stranded, could be double-stranded. And then bacteria have a single circular chromosome. That's their actual chromosomal DNA, and they could have plasmids that can carry other genes, like antibiotic resistance and stuff like that. So that's what I'm talking about. So our specific cells wouldn't have plasmids, but fungi and stuff could, protozoa could. we have mitochondria that create all of our energy. We just learned about that. They have their own mitochondrial DNA. It's circular. The picture is not accurate. It's actually circular. And then chloroplasts have their own DNA as well. Same sort of situation. Okay. Our chromosomes exist in our nucleus. And they're only an X shape whenever it's getting ready to divide in mitosis. We learned about that. So chromosomes. A chromosome, that term literally refers to a discrete cellular structure of neatly packaged DNA molecules. So this is what's coding for the basics, the basis of that organism, right? Not the extra genes that were introduced, but the stuff that typically comes with it, that organism type. So eukaryotes, we have our chromosomes in the nucleus. That's what it means. U is true. Karyo, we're talking about the nucleus, right? True nucleus. They could have a variety of numbers of them. So they have several. We have 23 in our 2N. So we have a copy from our mother and a copy from our father. So we have 46 overall. So it's just to let you know a little bit about that. So that would be what we call a diploid when it exists in pairs like that. And then when it's in singles, we call that haploid. Okay, and so the R's also, you guys have seen them in a lot of the pictures when we draw them in those X's, even if that is just when they're getting ready, but you can tell that's not a circle, right? It's linear. Bacterial chromosomes, however, are usually, they only have one, can get complicated, but they usually have one, and it's usually a single circular double-stranded DNA chromosome. Our chromosomes as well. Our chromosomes are DNA. Okay. Don't get confused with these viruses that are coming up. So genes, what does that word mean? We talk a lot about our genes and what they code for, whatever. Like, oh, you have the gene for blue eyes, and oh, you must have genes for being tall or whatever. These are basic, or you carry the gene for breast cancer, all that sort of stuff. These are basic informational packets. They carry a usually discrete bit of information regarding a certain aspect of that individual or trait of that individual. So when we're talking about like classical genetics and the basics of that, when we're talking about Mendelian genetics and all of that, we're talking about genes being the functional unit of heredity. Now, of course, things have gotten complicated because we've got these freaking viruses and all that. But that's the idea. The gene is the functional unit of heredity. It codes for a thing that does a thing predictably in an organism, right? In their cells, if they have cells. So we can break this down now as far as molecular and biochemical genetics goes. So then we would be referring to specifically a gene being a site on the chromosome that provides information for a certain cell function. So it contains that information. It could code for a cellular function or a trait or something like that. So not necessarily so general as saying a functional unit of heredity. Now we're talking about information. It's coding for information for a specific function. So now, what's the preferred definition? We would say a segment of DNA that contains the necessary code to make a specific protein or an RNA. And yes, that will have a certain function within the cell or for the cell. So it does get a little bit complicated, but it's not meant to be, right? A gene codes for something that does something for the cell. So we can break this down now. We can start talking about genotypes and phenotypes. The genotype is a sum of all types of genes constituting an organism's distinctive genetic makeup, okay? You could carry a gene for A blood, the type A blood, and you could carry a gene at the same time, maybe that's from your mother, and you could carry a gene at the same time for O type blood that you got from your father, but you would express the A. We would see the A, right? Okay, so you could have both going on there. The phenotype, the part that is expressed, the part that we would see happening is the expression of that genotype. The AO would be seen as an A in that. So this is what is kind of what is expressed right? So down here, what we're looking at is phenotypes versus genotypes. In this situation, we had a Mendelian setup. And what that means is Mendel's. It doesn't, don't get too much worried about what I mean by that person's name. But if we had two parents, and this was the mom, she had a big, why did it have to be P? Like you can't even tell like the letters, right, from each other. So let's not use P. Let's use B. So this was the mother's genotype, right? What the mother had from her mother and her father. She got one big B and one little b, one from her mother and one from her father, okay? Then we had the same thing with the father figure here, the gene donating people involved in this, okay? Okay, so these are the possible outcomes we could have. In general, in these situations, when we have capital letters like the big B here, that is typically the dominant trait. So the trait that is going to be expressed and seen, you know, it's going to be like even if you carry the other traits, you know, that aren't as dominant, they have to be the recessive traits is what I don't know. I could only think of repressive, and that's not what I was trying to say. Recessive traits, even if you carry one of those, that's not going to be seen. The dominant trait will be. And there's a lot of reasons why, but you don't need to know about it, right? So anyways, these are the possible babies they could have. Their baby could be big B, big B. So we're just kind of going down and matching them up, right? It could be big B, little b. It could be big B, little b over here, or it could be little b, right? And each of these is going to express something. So we do know that big B is dominant. You only need one big in order to do the big B thing. So in this case over here on the right, we're using B's instead of P's, but those big B's, the fact that they're there at all, those are the chance that you're going to see purple. So that's why in the phenotype, we see all this purple. That's what's being expressed by the genes, okay? Only in this one, like 25% chance case will we see white, where we had the two recessives come together. There's other terms we can use to describe that situation. When it's homozygous, that means that you have two that are the same. So we have two that's the same up there, and we have two that's the same down here. And then these ones in the middle are heterozygous. They have different, one is big, one is little. Those are some of the terms associated with the genetics there, but you guys don't need to worry about delving into that too much, I promise. Okay, so nucleotides. Nucleotide is the term referring to those subunits that make up both DNA and RNA. Okay, we're gonna talk about DNA first. DNA, those nucleotides consist of a phosphate, which is that a phosphorus atom, looks like a P, and it's surrounded by, and I'm not going to do the double bonds or anything, but it's surrounded by a bunch of oxygens. Those oxygens that are surrounding it are very important. It's giving it that negative charge and support group, like when we were talking about like ATP and all of that, for that push, that storing of like a potential energy there. Okay. Okay, moving on. The sugar for DNA is deoxyribose. So that deoxy here, we've removed literally an oxygen. That's all the difference is with the sugar for RNA, which is just ribose. And then we have the addition of a nitrogenous base. Nitrogenous just means it has a bunch of nitrogens. Nothing else. It doesn't mean anything else fancier, okay? There are 4 possible nitrogenous bases in DNA. They come in 2 forms, the purines and the pyrimidines. The purines are the double ring ones, and the single ring ones are the pyrimidines. Now, I'm not going to have you guys draw out or anything, but this is basically what it would look like. These are the double ring nitrogenous bases, and this is going to be dealing with the single ring. So see how there's only one ring over here, and then there's 2 kind of linked together over here. That's kind of what we're talking about, okay? Single versus double ring. So purines are the double rings. Pyrimidines are the single rings. Purines are going to include the A and the G, and the pyrimidines are the T and the C. Now, this might not help. I'm going to try to share it with you. When I write out A and I write out G, I feel like there's kind of more involved in writing those out than if I write down T and C. Those are simpler, right? So their actual shape is simpler. So if there's simpler shape of the letters, then I would think that this would be the single ring ones, and these would be the double. Now, the only way that I happen to remember, I have to go back, I have to start here kind of every time. And the only way that I ever remember which ones are purine or pyrimidine, because I can't remember that pyrimidine is a double ring or single ring. I always get confused about those. So when I'm like testing or studying for something like this, what I always remembered by is like pyrimidine. When we start talking about UV radiation causing like cancer and like mutations, we talk about pyrimidine dimers or thymidine dimers. Okay, so pyrimidine, that's what I always think about, because they kind of bond across on that side, the one side of the helix, instead of across to the other thing that they're supposed to match up with. They create the wrong kind of bonds. So we'll talk about those more a little bit later, but that's how I kind of remember it. So I know that T is going to be thymidine or pyrimidine dimers, because I've talked a lot in my past about, pyrimidine or thymidine dimers. That's just how I remember it. So I know that if the T is a pyrimidine, then the C, which is also a simple shape, is also a pyrimidine. So that's how I kind of remember those and just lump the other ones on the other side as far as pure. So it is that complicated in my mind. I'm so sorry that comes off that complicated to you as well. Moving forward, when we're talking about these letters, I talk about A, T, C, and G. A stands for adenine. You do need to be able to recognize these words. Guanine is G, thymine is T, and cytosine is C. Okay, so these on the right were our pyrimidines, and these on the left were the purines. But the A always pairs with the T, and the G always pairs with the C. Now, when I always think about, actually, that's Chargaff's rule. I always say Chargaff, and then everybody always makes fun of me. But Chargaff's rule, and it basically is going to say, A always with T, G always with C. That's how they bind with each other in the ladder. I remember this pretty simply because G's and C's look similar. So they're going to bind together. The G's and the C's, they're both round. They're going to stick together. So the A's and the T's are going to stick together. So that's how I divide those up. Okay. So which of the following is not a pyrimidine? We said pyrimidines were the T's and the C's, right? So we know that this is a pyrimidine, this is a pyrimidine, and I'm going to let you know right now, uracil, which we didn't talk about just previously, I don't believe, in a second ago, but it is also a pyrimidine. It is a lot like the T, but it's for RNA. Okay, so the answer here would be adenine. Okay, sorry, there is a washing machine going on upstairs. I don't know if you guys can hear, and I apologize for that, but I'm in my apartment, so I don't get a lot of choice about that sort of thing. So we're talking about anti-parallel arrangement, right? So we're talking about the direction that the DNA goes. Everything that we're pretty much talking about forward to backwards, freaking A, man, is going to be in terms of 5' going to the 3' at the end. That in general is the correct way to go. Okay, that's the way that you would read the genes and everything like that. Okay, 5' to 3'. The other one, however, is just the opposite. It's like the one is going in this way where the genes are and all of that, and the other one is just the exact opposite copy going in the opposite direction as well. So that one would look to us straight on as 3' to 5', but it's not actually going in that direction. It's still going 5' to 3'. It's just going the other way. Okay. What does that mean? Well, definitely know this, regardless of if you understand what it means. I'm going to show you, though. This is a huge deal as far as how DNA works. 5' and 3' is talking about the carbons. The actual, okay, let me go ahead and erase the carbons. The carbon number or position, where we start numbering from 1, which is here. You can kind of see it on this picture here. And then this is carbon number 2, this is carbon number 3, this is carbon number 4, and this is carbon number 5. So this is the very, very, very end, one end. And so you can see that the 5 is up here at the top. So this is the 5' end. And then if you go down here to the bottom, it'll end with that 3 sugar, that number 3 carbon in that sugar backbone, sugar phosphate backbone alternating. We're talking about the carbons for the sugars here. That is the 3' down here. So then we know 5' down to 3', and then if we looked over here on the other side, it has to be the opposite. So that we would have the 5 down here, and we have the 3 up here, and it will be moving in. Then therefore, it's always 5' to 3' in that direction. So that's what that's talking about. 5', 3', talking about the placement of the carbon, which carbon we're talking about in the sugar part of the sugar phosphate backbone of that molecule. Okay. You probably didn't learn a thing, but I hope that you did. Okay, so this is a difficult question. Clostridium tetani DNA has 14% Gs. What percent of its DNA is A? Well, if it has 14% G, then it has to have 14% C, right? Together, that makes 28 GC, right? So what is left over after that? Well, we've got 100% minus 28, which is, sorry, I'm really bad at math, guys. 72, okay? And then that's what's left over. So that would be A, T, right? So half of that is going to be A. So that is going to be 36. So that should be C. Okay, so that's how we did the math of that. It's so fun, right? Here's some things involved in DNA that you might want to know about. These are enzymes that are involved in dealing with DNA, right? And what they do. Helicase unzips it, primase makes an RNA primer. We'll get into why. DNA polymerase 2 and 3, making the new DNA chains and going in and filling in gaps and mismatches and all of that. Ligase is joining together pieces of it. Topoisomerase is making sure that it's not getting too coiled up and stuff like that. That'll get tension whenever you like unravel it, like you would with like the telephone cord or whatever. You start straightening it out in the middle, it's going to get really tight at the ends, right? And then there's another one down here, topoisomerase 2 and 4. 2, I guess we call DNA gyrase. I didn't realize they were the same thing until just now. But anyways, basically what those are going to do is dealing with any supercoiling and stuff like that that may have originated during this process. The ones that you probably should be aware of, I would definitely be aware of what this does, definitely what the DNA polymerases do as far as extending the chain. The ligase is important as well. The case is kind of more, it sort of takes care, it sort of tells you what it does, unzips the helix, right? All right. DNA polymerase 3, it makes new strand of DNA, daughter strand, using the parental strand as a template. So right, whenever I told you that A binds with T and C binds with G, it's gonna, if it like had a parent strand that we've separated their helix apart, right? Now we've got one strand that has A, T, G, C or whatever, then your copy would be T, A, C, G, right? And so that would be what the new helix would read. And you could do the same thing with the opposite strand, which would start out as this and then do this, make the copy into this. You have two strands that read exactly the same. One strand came from the original parent strand. This one below it was synthesized new, and the same thing would happen with the other side. Okay, so that's how that's going to work with DNA polymerase. So we have to unwind it all first because it is all in that lovely little ladder, the helix ladder that we're so used to seeing with, you know, DNA So that has to get unwound first. That is, we're going to unzip it open with the helicase after we unwind it, right? And we cannot add new nucleotides on. So we were talking about adding the T, A, C, G. We can't add on the T or the A or whatever, just out of freaking nowhere. We have to have something added on to the end of it, the side, right? Adding it on to something that's already there. So that is where primer comes in. It adds a little bit of something to add on to, okay? We can also only add from 5' to 3'. This is why I say this is the correct direction. We can only move from 5' to the 3'. Now, we'll see why that's a problem in a moment. So during all of this, we form this thing called a replication fork. That's where the helix has been unwound, but the rest of it on the sides are still wound up, haven't been unwound or unfurled or anything. So where the replication is actually occurring at that very moment is the replication fork, okay? So if we're doing this in bacteria, that's a circular DNA molecule, and we will have 2 replication forks, and it will meet up with each other. Eukaryotes have just a lot, okay? A lot. And so that primer that I was talking about, that gets you started off on making the copy, that's a little bit of RNA. It has to be RNA at first, because we're just going to get rid of it eventually. It's sort of temporary. Think of always RNA as being the temporary thing and DNA being the permanent thing. So our leading strand is the strand of DNA that we can make just all in a straight line, all happily in 5' to 3' direction. Just start with our primer, of course, and then just go all the way down. Then we have the lagging strand. I'll show you a picture on how this makes sense in a moment. Well, then we have the lagging strand, and that's essentially going to have to be made in short segments because we can only still go in 5' to 3'. Okay. Those little segments that we would be making to keep going in 5' to 3' with the opposite strand, the segments as we go, those are called Okazaki fragments. And they're synthesized in 5' to 3'. And then we'll have to seal them all together with ligase to make a continuous strand. So like I had said previously, the new product that you're going to have made, one strand, like if this was your parent strand, right? And then we separated it out so that this was your parent, and then this was a parent, and then we wanted to make new, then this one would be a new, and this one would be a new. And then those together are new, right? That's our resulting daughter strands, but part of it's from the old and part of it's from the new. So that's semi-conservative replication. All right, this is what it's going to look like. And this is where you'll start to understand what I'm talking about with regards to the actual Okazaki fragments and everything. All right. So the top parent strand, we are able, so this is on this one, starting with a 5 going in this direction, right? Moving 5' to 3' on this direction, we can just go continuously because we have to move 5' to 3'. As it opens up, we continue to open up more space. But we cannot move on this new strand, which is the top one. We cannot move in this direction because that's 3' to 5', and we have to move 5' to 3'. So what we do is we have these fragments that we create, and we start at 5' and move to 3'. So we add on our stupid primer because you have to have it first, and then we move in the 5' to 3' direction and make up for whatever space was made up by opening up this fork further, okay? And filling in those gaps gradually with these little pieces that we call Okazaki fragments. Then DNA ligase, it's another enzyme that's going to come in and just seal that little, join it all together so it's continuous. So that's how that works. Because you see here on this left side how that we would be opening this like a zipper. And we can just keep going with this one, but this one we have to still go in that direction also with the opening, and then come back in with our RNA primer once we get enough space in there so we can still read in 5' to 3' direction for that strand. because this thing is still going to only ever open. Like I was just saying, still only going to keep going in that direction, opening the fork in that direction. So you have to still do it by pieces. Let's just keep saying the same thing over again. I apologize. Right. So this is just more words on describing how that happens. We'll be opening up the helix via helicase, and there's other proteins that are getting involved in all that. But basically, the DNA polymerase is reading along in its 5' to 3' direction. And as it opens up, we'll pop on an RNA primer and then read in 5' to 3' direction for these pieces called Okazaki fragments that we can then stick together with DNA ligase and make a continuous strand. This is for eukaryotic DNA. You can see the replication process is the same, essentially, but we will have also things going from both directions, but multiple origins. So we'll have a ton of these going on, not just 2 meeting up in the center of the circle. We'll have a bunch going on in that whole linear piece because it tends to be a lot larger. And then that's where the topoisomerases come in to keep everything from getting too strained on the helix itself. So basically all of this, we have DNA, we just talked about DNA and how to make more of it, which you would need to know if you're making new cells. So how do you copy that? And then we talked about what a gene is. It codes for basically a functional thing for the cell, right? So how do we get that to happen? Well, then we go through this thing. We always considered this central dogma that viruses just pooped all over, essentially. But we start with DNA to RNA. And that is transcription. And then RNA will read the message that it contains and make protein. So that's called translation, that process is. We used to think that could only happen that way. We can only go DNA to RNA to protein. We now know that we can go back this way at least. That's at least that. So viruses told us that. And it's a useful thing to know, but also terrifying. So sticking with the basics of what we just learned, DNA to RNA to protein, okay? We're going to talk about the details of how we get through all of this. That was just a chart showing you the general thing, what we're getting ready to talk about. So the DNA is going to tell us a gene that will determine the protein structure, essentially. So proteins are essentially what is going to, the results of, so DNA goes to RNA, it goes to protein, right? So that tells you that if your DNA, all of that, just it only codes for proteins. Okay, that's all it does. So that means that everything about you was made by something to do with a protein. Now, maybe not all of it's made of protein, but a protein had a hand in it, probably. Anything that determines anything about you, a protein probably had something to do with it. Cool. So that's what I was trying to get at. So these genes carry the information for that protein's sequence, exact amino acid sequence, and its shape and all that, therefore its shape in that, because that's determined by the sequence. And the phenotype is ultimately determined by those proteins, right? The protein expression. Proteomics is part of science that studies the complete set of expressed proteins in an organism, all the expressed proteins, like everything that it actually makes. So DNA is mainly a blueprint. It's a big, like a blueprint for an entire house that tells the cell which kinds of proteins to make and how to make them. So anytime that you need a protein, you would go to DNA, pick out the part that you wanted, like we said, the bedroom or the kitchen or the whatever, bathroom or whatever, pick out the bathroom one, and it would say, okay, this is how you put the bathroom together. That is the RNA, the mRNA, the messenger RNA. Okay, let's get into it. So this is going to show us DNA essentially. So if this was our 5' to 3' and whatever, 3' to 5', essentially, okay, just to remind you guys about it. But anyways, you have 2 strands. These exist with a bunch of our nucleotides all in a row. If we divide it in 3s, in triplets, each triplet will correspond to an amino acid, essentially, at the bottom. So the triplet, that's in the DNA, we call the triplet. Okay, so these 3 will essentially code for these 3. Now, this is the codon for the RNA. So it's just the triplet for RNA. That's the word that we're using for the mRNA, the messenger RNA. Okay. We made a copy of that top strand. This is what it looks like. This triplet here, or sorry, this triplet up here became this codon for the mRNA, the messenger RNA. And that codon is going to be what's important for deciding the amino acid assigned to it. And then all of that is going to lead to a bunch of amino acids that are going to get linked together. That's going to make up the protein, the primary structure. They'll fold up certain ways based on the chemical interactions for the secondary, and then yet again, tertiary, and maybe even quaternary if we need multiple units. That's how all that's going to work. Important to know with all of that. We just saw about the messenger RNA. This is the tRNA. What does it stand for? It stands for transfer RNA. Transfer RNA is RNA, just RNA itself folded up. But what it does, it has an anticodon, which is important here, right? And that anticodon, we'll see on the tRNA, it has UAC. So U is like a T, right? So U is going to pair up with an A. So this is our messenger RNA, AUG. This transfer RNA comes in and matches up that codon and reads up that codon, those 3 nucleotides of the RNA, and it comes up with its complement. So A binds with U, A binds with U, but U with A this time, and then G with C. This is the complement to it, right? And based on that match, this guy up here is attached to a certain amino acid that corresponds specifically to those 3. That's how it works. And then this is how the ribosome would function. The tRNAs come to each part and will read codons here and then attach certain amino acids that they're carrying around to one another to make entire proteins. So we need to make the RNA, the messenger RNA first. It's kind of similar to what we had with the DNA, but we're making RNA, right? So we use U's instead of T's, right? Uracil instead of thymine. So we'll unwind. RNA polymerase will come in and recognize a portion of where it needs to start. That's the promoter region. Okay, that's where it needs to start. Polymerase means poly making, taking a lot of the MERS and making polys out of it. So it's just making RNA out of its appropriate nucleotides. So that's what polymerases do. DNA does appropriately DNA nucleotides. So we're going to start by making the complements to that, and then we'll get the mRNA matchup from that DNA accordingly. So A's will give us U's, and G's will give us C's, and all of that will match up accordingly. Okay. So this is the template strand. That's because they're going to have an opposite read to them. So, T on this one will give an A on this one. But over here, if we looked at the same spot on the coding strand will read the same thing as the mRNA. Okay. Anyways, that's what's going to be going on. We unwind, we stick that RNA polymerase on, and we can just start at the promoter region and start adding on ribonucleotides accordingly. All right, so this is a picture of that as we move on. Again, 5' to 3', using nucleotides that are around in the area, which is called the nucleotide pool, whatever's around there, and just keep going on until basically to stop. So we'll get to the end, blah, 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 blah. We're making longer mRNA. It could be thousands long. Okay. Making mRNA. Let me turn the captions on first.
During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other.
Okay, cool. Right, moving on. Okay, the message that is contained in messenger RNA essentially is a part of the gene that's actually going to code for a functional, usable protein. Okay, that's the idea of what mRNA is. So we will read mRNA in codons, which is a group of 3. We're talking about the mRNA, it's codons. We're talking about the DNA, it's triplets. Okay, so the codon will tell us which amino acid is going to be added, and this is pretty universal. All right, so this is the code that will code for everything ever. If you get UUU triplet, then codon, it will be, TTT, but you know what I'm saying. UUU, it codes for phenylalanine. If we go down here, and you would find the first ones on down here, the second ones will be up here, and the third ones, if you need to, is over here. Now, so we'll start, I don't know, if you had CGT or CGU, we'll find the C in this line here. So anything on this line. So we said CGU, so we'll go to CG, and then you can see the U here, right? So that's going to code for arginine, and that's how it works, right? So the transfer RNA is going to match up with using the opposite code and match up with those codons, and give us the correct amino acids associated with them, and then they will get bound together. And when those amino acids are bound together, we'll create the protein that the gene coded for. Okay, so there is some redundancy, obviously, whenever we look at this. So look at proline. Any of that can code for proline. Any of that can code for threonine. So there's some redundancy there. Certain amino acids are going to be represented by multiple codons. So that will allow for insertion of the correct amino acid, even if there's a mistake in the DNA. So that's trying to protect from that, because there are going to be mutations. We can't avoid them all. And then wobble is a term that's referring to the first 2 nucleotides being important, right? Because really it's the first 2 that kind of determine everything. And then the third one, it's kind of a gimme, right? So if we have CA and then CA down here, we can see that we do need the U or the C, but it's the U or the C here. It has to be the U or the C. We've got 2 choices here. So it's a little bit different. For the most part, it's going to be, UC is going to be serine, and then AC is going to be threonine, and whatever. CC is going to be proline. You guys get it. So that's what wobble is talking about. So some of them have more wobble than others. But that's the idea. So that allows for some, again, mutation and things go awry. You guys are so snoring. Freaking little dogs. Sorry, I had a book to throw. Anyways, right. So back to what we were saying. We've got DNA, and in the DNA, we will have triplets. That's our 3, right? If this is the code, if calling this is the coding strand, then our mRNA will match it exactly. It's not the complement. It's the mRNA's complement to the template strand. So if it's ATG, the copy of that for our mRNA will be AUG. So that's what this is saying on here. And then our tRNA will read UAC. The U will like to bind with this. I don't know why that went away. The A will like to bind with this U, and the C will like to bind with this G. Okay, so that's how the tRNA will interact with that. And based on that interaction, when reading the mRNA codons within the ribosome, we will get a product of a certain amino acid. In this case, it's F-methionine, which is the first one, always. Okay, so all of this has to happen in order to make protein. We will need mRNA, copy of the DNA. We have to have tRNA to recognize the codons with the anticodons. We have to have amino acids linked to them that are appropriate to match up accordingly, and then ribosomes to read it. So this is all for translation. So we'll have initiation, elongation, and termination. Pretty straightforward set of steps for a lot of this stuff, right? So this is pretty wordy explanation of what is going on in these pictures. It is kind of what I was telling you as far as you have your mRNA divided into codons, which are units of 3 nucleotides for the RNA. The tRNA is going to match with the complementary matching for those 3 with the anticodon. If the anticodon matches the codon, then that amino acid associated with that anticodon, and therefore codon, will attach to the protein that it's making. And they'll keep going and going and going and attaching. Okay, so that's the idea. You can read through this if you would like to. I think it's a lot of words. We have until it gets to a stop. The stop literally just tells it we're done. Okay. There's a video. I think that'll be more useful. That's what I'm trying to get to.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA, paired with the previous codon, is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
I think the best way to try to kind of study all of this and try to understand it is to try to write it out for yourself. Don't copy it. Write it out as you are understanding it, and then go back and check over and see what you're missing and what you need to relearn, and then keep doing that until this is not a touchpad, until you kind of get it down, because there's a lot in these chapters. So anyways, moving on. So protein synthesis, we have a start codon, literally says this is where we're going to start with our amino acids. It's first 3. We have a stop codon. These are what we call nonsense codons. They do not code for an amino acid. They code for nothing except for stopping. Causes it to stop making protein altogether. No more translation. And then we have translocation. This is where we're moving that ribosome down, and we're having as we're shifting over those tRNAs. Okay. So we already looked at this, but just going back to it so you guys can see kind of how it would be moving and how I need to jump back and forth and read it and everything like that. Okay, so we made our protein. Some proteins, that's not the end of it. A lot of them, it's not the end. You might want to add certain groups onto it. That could be, you could be wanting to clip off that first one, which is always formal methionine. You could be wanting to add sugars or fats onto it or something like that. You could want to add other cofactors, like iron, if we're talking about hemoglobin, or other things that are, like coenzymes for making the apo enzyme, the holo enzyme, right? So anyways, and you'll need maybe more than one subunit to come together to make a functional protein of some kind. So there's more that goes on after that sometimes. So with eukaryotes, so that was all bacteria. So that's what we talk about the most in this course. That's bacteria and archaea. For eukaryotes, we have a different kind of methionine at our start. It's not that formal one. Doesn't matter what it is, but it's different. Okay, so that's one of the differences going on there. Also, eukaryotic, our mRNAs will only code for one protein, whereas bacteria will like link an mRNA. The mRNA will have a whole bunch of proteins linked with it. Like you'll code for a whole bunch of them because they're usually involved in one process. You'll need all of them. So that's why they're linked that way. Eukaryotes also have to be a little bit freaking ridiculous, complicated as well, because we have these things called introns, and that's parts within that gene sequence that aren't coding for what we need at that time. So we will clip those out literally with an enzyme, a special enzyme for it. This is the introns, the greens. So we have an enzyme that just cuts it out and re-stitches together the ones we want. The ones we want that are coding, having the coding information on them, those portions are called exons. These are the coding regions. Okay. Viruses are complicated. They can have a lot shoved into one genome, and each of these is going to be like a protein, but they're very efficient and very compact with a lot of information in one spot. Remember, it can be DNA, it can be RNA, it can be single strand, it can be double strand, it could be the 5' strand with the coding strand, or it can be the template strand, the 3' strand. Okay, so complicated. What structure is actually doing the translation process? That thing that's actually doing it is called the ribosome. Okay, moving on to operons. These are only bacteria and archaea. We don't have these in our own eukaryotic cells. Okay, these are prokaryotes now. Operons can be inducible, so there could be something there that makes it start working, or they can be repressible. We talked a little bit about these concepts in the previous chapter. So if you guys need to go back and kind of refresh your memory on the inducible and repressible enzymes, this is, and like how they're regulated with like products and stuff like that, this is definitely related to that. So remember, this is chapter 9, so that would be back in chapter 8 in the metabolism one, the very, I'm sure, not convoluted at all one, metabolism. Okay, so that might help if you're a little bit confused about these. All right, so these, whether we're going to be inducing or repressing, is going to be determined entirely how the environment is affecting the cell. And it will determine whether or not we're going to start or stop with something. So the inducible ones, the ones that can be turned on, these are usually related to catabolic reactions. So what do you guys remember what catabolic is? Breaking molecules down, right? Enzymes that act in catabolism are involved in breaking larger molecules down into smaller ones, usually to make energy or serve as nutrients, so building blocks and other things. So this means that the operon will be turned on by the substrate typically. So whatever we are wanting to break down or use. So if we had operon that was involved in, I don't know, glucose, because we talked about glucose previously. When glucose is present, it will come and interact in a way that will turn on making enzymes that will allow for breaking down the glucose and running it through like glycolysis, for example. Okay. So often we have enzymes that are needed to metabolize that nutrient in this case, they're only going to be made when the nutrient is present, is actually there. We're not going to make all these enzymes for breaking down like glucose. If glucose isn't present at all, that's a waste. So moving on to the repressible ones, that's the catabolic reactions previously. These ones, inducible operons have to do with breaking things down. Catabolic, we're not going to need those until the thing is present. Repressible ones are going to deal with the anabolic, so building things up. So this is usually going to be involved with genes in a series that are related being turned off or repressed by the product. Okay, so the enzyme is making a product, and once we have enough of it, we can go back and turn that off. So we're going to start with the lactose operon. With the lactose operon, we have a regulator, which is the gene that codes for the repressor. Okay, the thing that's going to repress it and stop it, right? Then we have the control locus, how we're going to control all of this. The promoter, which we just said previously was what's recognized by RNA polymerase. So that's where we're going to start our mRNA being made in transcription. We're going to transcribe it, right? And then we have something called the operator, which is kind of the on-off switch, right? And based on whether we can do that. Oh my gosh. Sorry, go back to this because I'm getting a phone call. Okay, sorry, I'll pick up where we left off. Then we have the structural locus, and that's just referring to the genes that are going to make the enzymes for this, okay? The genes that are necessary for moving all this on. Okay, so this is what it's going to look like. The regulator gene is going to make the repressor, right? So this is brown hook, like horseshoe looking guy here. All right, so what's going to go on initially is this operon is normally off. Normally this is not doing anything. We're waiting for lactose to turn us on. That's what this is about. So what happens is the repressor sticks on over here by the operator so that when the promoter, this over here, when the polymerase sticks onto the promoter where it's going to start making mRNA, it can't go because that is there. The repressor is locked it up, can't move. All right. So that's how it's off. To turn it on, when we have lactose introduced to the system, our inducer, because this is an inducible operon, we are going to have lactose interact with the repressor. When that happens, there's a chemical interaction, right? Because it's all chemistry. It's going to change the shape of the repressor. So it's no longer going to be associated with that DNA. This is the DNA where this is all happening, okay? So now the RNA polymerase can come and interact with that DNA area and start making the mRNA, start transcribing it, right? So that's how we would turn it on. So we will transcribe. That means we're making mRNA. We're putting that mRNA to use with some ribosomes and some transfer RNA so they can match up with their codons of the mRNA and the anticodons of the tRNA, and the amino acids can link together. And all these genes will make all their own individual proteins. Now we're making all these friggin' proteins up, and now we can start breaking down lactose, which is the goal. We had lactose, now we can break it down. Catabolism. Catabolic things going on, reactions. So we're breaking it down. Okay, so when we are depleting lactose and we've broken all of it down, there's no lactose around anymore in the environment. When this is all gone, eventually it'll dissociate here as well, and that repressor will stick back on and lock back on until it comes back again. That's the lac operon, okay? All right, so that was an inducible operon. Now we're going to talk about repressible operon. Repressible operons are in the on mode and will only be turned off when whatever it's making is no longer needed. Okay. So the excess nutrient serves as a co-repressor in a way. But we're going to show you what I mean by this. Okay. We have our promoter region where our RNA polymerase is going to stick onto, and our operator, which is going to be the important area where the repressor gets involved. All right. So normally it's just on, right? So we can go through all of these genes that are going to make up whatever we're trying to make. Cool. We make our mRNA. It goes through the ribosomes with the tRNAs. It matches it up. We make our little genes, our proteins as a result of all of that happening correctly. And now we have made enzymes. Over here it's telling us enzymes, again, proteins that are going to synthesize arginine, which is an amino acid. So we have that going on. When there is an excess of arginine, because normally this will be used immediately, okay? When we're not using it immediately, we don't need it as much, or we have an excess of it that's been made, then it'll come and interact with whatever its repressor is. Again, repressor in general apparently looks like brown upside down horseshoe. The arginine is going to interact there and cause it to bind on the operator. So we will stop making the enzymes that make arginine. So that is how we control how much arginine would be making in that environment, in that repressible operon. So that's usually, like we said, anabolic reactions. So we're making arginine. So we're making something. Okay. Antibiotics can affect this stuff, stop it and inhibit it and whatever. Some drugs that inhibit protein synthesis, those steps, rifamycin, actinomycin D. Then there are some drugs over here on the right that interfere with the actual ribosome's function itself, like erythromycin, one that you might have heard, chloramphenicol, tetracycline, aminoglycosides. Those actually interfere with the ribosome, so it can't function. So that's kind of a neat way that functions. Just remember, our eukaryotic ribosomes are the ADS. The eukaryotic ones are ADS, while the prokaryotic ones are 70S. So they're a little bit different. So the structures are going to work a little bit differently. So the repressor protein, where does it interact with that whole system? Do you guys remember what it was? Or the promoter is where the RNA polymerase is getting involved. The operon is the whole set of genes and all of that. The promoter, the operators, and all of that. The exon is involved in eukaryotic things. This is the eukaryotic coding portion that is what we want to keep from a gene, whereas an intron we cut out. So it would be the operator. This is the operator of the operon. That is where the repressor is going to interact. Okay, recombination. That's whenever bacteria donates DNA to another bacterium and it gets involved in its whole, how it works, how it makes things and everything like that, how that organism lives. Okay, so after this happens, we get new DNA involved. We have a new strain, essentially, a strain that is different from both the donor and the recipient, because the donor wasn't quite the same originally. You know what I mean? Like they were different originally. So now we have a new strain going on. And that's what's going on there. A lot of times this happens with plasmids, those little extra DNA circles that can code for things like antibiotic resistance. Right. So recombinant is a term for the organism that expresses genes from that it got from another one originally. It's not part of its original makeup. It got it from somebody else. Okay. That's a recombinant, a genetic recombinant. This is talking about genetic recombination. Horizontal gene transfer is going to be any transfer of DNA where the organism requires new genes that did not come directly from the parent, not from a parent, is horizontal. Otherwise, it would be kind of vertical, is essentially what we would say, right? Transmitted through heredity. This is non-heredity. Hereditary. Good movie. Okay. So moving on to horizontal gene transfer and what's going on there. Remember, we said plasmids, important little pieces of circular DNA that can replicate independently from the chromosome itself. And we can move it between bacteria, and it can code for things that help the bacteria live. So that's not necessary, but can help it live better, more easily for it, right? So we can transfer DNA between cells via plasmid. We can find these in most bacteria and in some fungi, some protozoa. They contain at most a few dozen genes, not a lot, right? So we have 20,000 in our chromosomes. This is just a few dozen. And it's not necessary, like I just said, but it can have useful traits like antibiotic resistance, right? So there's 3 ways we can do this, either through conjugation, transformation, or transduction. So we're going to go over these. I don't want to talk about it here. So conjugation is like bacterial sex. We're talking about exchanging genetic information via maybe plasmid or part of even your own chromosome or whatever. But we take part of it from the donor, the starting cell, and we give it to the recipient by a direct connection, usually through a sex pilus. That's conjugation. So literally the cell is moving it to another cell. Okay, so both of them can do this, gram positive or gram negative. Say both, there's other kinds that are out there, but gram positives or gram negatives can do this. But yes, this, an example of how they might do this would be via sex pilus. Okay, so we have our pilus being made here. We have our little plasmid going on here. We make a copy of our plasmid. Remember, they can replicate independently. And then we're going to feed that replicated version down into the new recipient. So that is the version of transferring the F factor. So conjugated plasmid. There's another version over here where the genes might be part of the chromosome. And as we make the whole copy of the entire chromosome, we might feed part of it down into the new cell that we're connected to by the sex pilus, and part of it makes its way down in there and can become then incorporated, that whole mess broken off and incorporated in there to the new one. So that's another way that that can happen. So it's not only plasmids. Plasmids are the ones that I want you guys to be most concerned about. Okay, resistance plasmids or factors, okay? These have genes that will allow whatever that bacteria or whatever is to resist antibiotics or other drugs, in a way allowing that bacterium to produce whatever enzyme it needs to break down the antibiotic or other things that could be involved, okay? They're commonly shared through that conjugation, the bacterial sex, direct transfer, and they can confer multiple resistance to a lot of different antibiotics. They can. And so they can also carry genetic codes for resisting, the presence of heavy metals like silver or something like that. And then synthesizing actual virulence factors like toxins or other aspects of the cell that could be helpful to that organism in surviving in the host. All right. The next one, we had conjugation, was, bacterial sex. Now we have transformation. This is literally getting DNA from outside of the cell. So here we have a bacterial cell, and then we have small fragments of DNA, soluble DNA just hanging out, and we're going to take it up. Cells that are able to do this and accept that genetic material are called competent for transformation. We do this in science a lot, and you will do this in the lab. So essentially what we'll have is original cell, bits of it's dying, it's breaking up, it's whatever. It's DNA is just out in the environment. This other cell, the recipient, takes up that DNA, that foreign DNA, and incorporates it into its own genome. That's an example of transformation. We can also do this with plasmids, with whole plasmids. So I'll have you guys watch this video about transformation real quick.
DNA transformation involves the transfer of naked DNA into a recipient cell. In the first step, double-stranded donor DNA binds to specific receptors on the surface of a competent cell. One strand of the donor DNA is degraded by nucleases while the other strand enters the cell. The single-stranded donor DNA pairs with an homologous region on the recipient DNA and is integrated into the recipient genome by a breakage and reunion mechanism called homologous recombination. If there are any differences between the nucleotide sequences of the donor and recipient DNAs, the mismatch repair system comes into play. The repair system removes either the donor or the recipient strand and replaces it with the complementary sequence. Since either strand may be repaired, some cells contain the new donor DNA and others have the original DNA sequences. In the laboratory, cells are plated on selective media so that only the transformants will grow.
Cool. What are you doing? All right, so transformation, I guess Frederick Griffith is the one that discovered this. You know, we're trying to focus on a little bit more on these individuals that were important in science. So there is that information. He worked with encapsulated and non-encapsulated Streptococcus pneumoniae, and the encapsulated strains have a smooth, and the ones that didn't have a capsule had a rough appearance, right? So essentially we could take the smooth S bacteria, put it into, the capsule ones were deadly, put it into a mouse that died. But when you take the rough ones that didn't have a capsule by themselves, the organism, I crossed out like it died, but what I meant to say was that it survived. So no sick, right? So it survives. Okay, so what we can do is kill the S strain. This rat, this mouse survives. It's dead. So what's important to it has something to do with it being alive, right? But then we can take that heat killed strain, put the rough ones that previously they did cause no effect. If you put it with the mouse survived just fine. If you put this S that's been killed, put it in the mouse, it survives just fine. But now we put them together, and what happens is the mouse dies. And why is that? That's because the DNA from the smooth, which has the capsule, which is making it, you know, deadly or whatever, has transferred into the rough ones and made them smooth now, or at least have that trait to make them deadly. Okay, so that's how transformation works. Now we're moving on to the third one, transduction. Transduction is whenever we have a bacteriophage, right? So bacteriophage, remember, that is a virus that infects bacteria. They can take their DNA, when we were talking about the lytic versus the lysogenic cycles, right? If we're going through the lysogenic cycle, we can have our viral DNA being inserted into the whole bacterial chromosome, right? And when it does that, it can insert genes as well that can give, you know, resistance, or they can give like virulence factors. So that can be going on as well. Okay, so when this is happening, whatever bacteria this is going on with, in this transduction, it has to be the same species as like what was infected before, because bacteriophages are usually pretty specific. So there's that. Okay, so then we have generalized transduction, where we have, again, this is with viruses, with bacteriophages. The random fragments of disintegrating host DNA are taken up by a phage during assembly when the host cell is not doing so good. We're making a ton of these bacteriophages. The phage will incorporate the host DNA, and any gene that was incorporated in that can be transmitted to the next bacterium. That's generalized. So you can pick up pretty much anything that way. Okay, so this is just showing that, a picture of it. So we've got the phage coming and infecting. We've got a piece of the host DNA hanging out. One of the phages incorporates it into itself, and then it goes to the next bacteria to infect it. And now we've put that host's DNA into that other new host. So that's what's going on. Then next we have the specialized transduction. So a very specific portion of the host genome is regularly incorporated because of where it normally will insert itself into the genome can be very specific. Then it can, when it is coming back out of it, the prophage is coming out, it will carry with it a lot of times the same genes every time to be predictable. So it's specialized transduction, same idea as the generalized one. It's just a specific genes this time. Okay. All right, lastly, I'm not sure if there's more after this, but now we have the transposable elements, something else that can be interesting as far as shifting around DNA and making things different. These are what we call jumping genes. Transposons are jumping genes. Essentially, literally, those genes can move around within the chromosome of the organism. That can lead to activation or inactivation of associated genes where this is happening, okay? And so what we would see with bacteria, there's a couple of instances where we might have the transposon here that could move to a different part of it, or it could break up and move, its parts could move elsewhere, or it could even, jump to different places, or it could move into a plasmid, which is going on right here. And then that plasmid could go and move into another bacterium, maybe through the pilus or conjugation or something like that, right? So those are transposons. All right, moving into the mutations. Mutations are literal changes in the genetic code. That's what a mutation is. Whatever it's caused by, it's the change in the genetic code. The actual literal nucleotide sequence in the actual genome, that's a mutation. This was the driving force of evolution, we believe, that as these changes occurred, some of them proved advantageous, and those with the advantageous mutations survived to pass those on to their offspring. In microorganisms, this is definitely the case, right? So this can become evident very quickly as far as altered gene expression can go with like altered pigment production or development of drug resistance. That can happen very quickly in organisms. We're not introducing any new DNA or anything, just mutation is happening on its own. You can see it happen in a matter of days. So a wild type is referring to the original, the natural non-mutated version. So this is usually the trait so we're talking about specific traits here. Wild type of that trait or gene, this is the natural non-mutated, most common, highest number one version, okay? And then the mutant strain is when an organism has a mutation that deviates it from the wild type. This could be spontaneous, it could happen randomly, just from errors, normal errors that happens, or we could have induced mutations that could be caused by something from outside affecting it. So those are called mutagens. Mutagens cause mutations, they induce mutations. Chemicals can do that. We have nicotine and stuff like that. Or we could have like ultraviolet radiation, that's a mutagen. Okay, so point mutations are little mutations that only affect one single base in a gene. This could be an addition, could be adding a new one in, it could be deleting one, it could be substituting it for something else. Those are possibilities there. Lethal mutations are those mutations that cause the cell to not be able to work properly and cause death of the cell, or be just totally dysfunctional and not useful at all. Then we have neutral mutations that have no real obvious effects at all. Those are possibilities as well. So what are some kinds of mutations that can occur? What we would see as the actual cause of the changes? If we have what is called a missense mutation, missense is changing. We have changed the genetic code. So now we have a different amino acid. Didn't stop it altogether, didn't totally shift it or make it bad or whatever. We've changed an amino acid. That can have a profound effect on the resulting protein though, okay? But the point is, missense is a change leading to a different amino acid. Non-sense mutation makes that protein pretty much non-functional, okay? It will change the normal codon of whatever it is to a nonsense codon, which we already mentioned were stop codons. There's no amino acid, and we can stop that production way earlier on in that protein than what we might need to be stopping it at, and not even get a functional protein. So that's pretty serious. Then we have a silent mutation, because you guys remember how in those amino acid things, I don't want to go all the way back to it, but you could have like CCC and like CCA, and that like coded for the same amino acid. Then if we change C to A, that is a mutation, but if it still codes for the same thing, the same amino acid. So same amino acid, that would mean that it is silent. We did have a change in the actual nucleotide, but it's a silent mutation. All right, and then we have what's called a back mutation, where we're literally going back to what it should have been, the original before. Okay, so it's reverting or reversing to the original. Okay, frame shift mutation is another one that I want to talk about. This is the big daddy of them. Okay, whenever there is some sort of base, a new one introduced or one taken out, that shifts the entire reading frame and changes all of the amino acids from then on. So if we inserted bases with this frame shift, because I was talking about inserting and deleting bases, if you insert bases in multiples of 3, it's not going to change the reading frame, right? It's still 3 is how many nucleotides there are in, a triplet for a codon. For an amino acid. So it's not going to shift the reading frame. It might add an amino acid in the middle, but you could have a still functional protein. You could. So it's like 1 or 2 insertion or deletion that's really going to cause that issue. Not in multiples of 3. So this is talking about those different kinds of mutations that we could have. So we have on here the missense mutation. So we have a T instead of what was supposed to be a G. Was supposed to be a G. So now we're going to be coding for this instead of this. Okay, so that's a missense mutation. All right. Now moving on to base substitution. This is going to lead to a silent thing. Every time. We did have A, so this is the silent mutation. We changed it to A instead of the G. So it was CTG, now it's CTA. And that's going to lead us to having ASP still. So it's the same. So there's no change at all. But we did have a mutation in the DNA, actual DNA code. But stop it. But we survived it. We still have the same amino acid there. So it's not going to cause any actual change. Now this down here, this is a frame shift mutation where we took out what was supposed to be TGGC. We have TGC. We've lost the G out, and now everything's moved over by 1. And now we're completely coding for other things. So that is whenever we had a deletion. And here we're going to have an insertion. So TGG, and it should be C next, but we're inserting a G here. So now we're going to be making these amino acids completely different from what was supposed to be going on up here, which is what was normal, what's expected. Okay, so that's how those are going to work. So what type of mutation would be shown here? So it's normally ATG, and we've now changed it to ATC. ATG codes for methionine, and ATC codes for isoleucine. It's a different amino acid, right? We had a change, makes a different amino acid. It's not a stop. Stop would be nonsense. It does change the amino acid, so it's not silent. That's the same amino acid. We can't tell. Yes, we can tell, but I'm assuming that this is just a change from ATG into ATC and not an insertion or deleting that's happening here. So we really don't have enough info for this, but we'll go and say we don't think it's a frame shift. We can say that it looks like it's a missense, if that's all that's happened here. Okay, so repairing damage. Why is this not going away? There we go. Repairing mutations. When you get one, how do we do it? There is something called photo reactivation. We have damage that's caused by UV, and we have ways to deal with that, right? We also have excision repair. So mostly we'll be talking about bacteria, I do want to be clear, where we can cut out the bad and put in the correct. Okay, so let's see a little bit about how that works in this video.
When cells are exposed to sunlight, radiant energy can damage the DNA. For example, ultraviolet irradiation causes covalent bond formation between adjacent thymines on the same strand of DNA. When DNA is damaged in this way, it cannot be replicated or transcribed. Most cells, however, are able to repair damage to DNA. For example, an enzyme called photolyase can break the covalent bond between the thymine dimers. This enzyme requires visible light, which is used as the energy source for bond cleavage. Hence, this mechanism is referred to as light repair. A second repair mechanism involves an enzyme that excises the damaged segment from a single strand of DNA. This process is termed excision repair or dark repair and does not require visible light. The excised nucleotides are then replaced by DNA polymerase. DNA ligase forms the final phosphodiester bond.
Okay. So that's what I was talking about with like the thymidine dimers and all of that. So that's what I always think of now. Okay, so mismatch repair. We're here talking about a repair system to locate mismatch bases, like a G didn't go correctly with a C and said there's an A there or something like that. So that allows us to go back and fix those because we didn't see them in proofreading. But we want to do that very quickly as soon as we possibly can because it won't be recognized if the repair enzymes don't see it. So this is the something called the Ames test. We use this to detect carcinogenic potential. So carcinogenic, we're talking about things that cause mutations, mutagens. So essentially we can use bacteria because they have DNA also and expose them to things. And if they get weird mutations, and we can do a much quicker sequence of their entire genome than we could of a human one too, on top of that. We can also look for actual change or lack of survival or whatever. So we can look at all of that from bacteria because it grows so quickly and see how mutagenic or how carcinogenic something could potentially be. Mutagens, Ames test, we're just exposing bacteria. They just think of it this way, exposing bacteria to mutagens and seeing if it causes any effect on the bacteria being able to grow. And if it does, then we wouldn't want to expose people to it. Mutagen, bad, right? Okay, positive and negative effects of mutations. Many mutations aren't repaired, so they're just going to be carried on in the cells of that organism. So that happens, right? Sometimes that can give benefits and sometimes not. It depends on the mutation. So mutations are permanent and heritable. So they can be passed on to offsprings, and some of that can be a good thing. Sometimes a bad. And then we have a small number of mutations that contribute to success, right? So that's what we were kind of saying. It doesn't always help, but sometimes it does. And when it does, those strains that were able to live a little bit better will overgrow the ones that didn't and pass on that more preferentially, those changes. So this is what we went through. I know it was a lot. We talked about genotypes, which is the actual, chromosomal makeup, what's in your genes. To your genetic DNA information versus phenotype, where that's what's expressed. We talked about what DNA is made of, those nucleotides, its structure, and how we replicate it. We talked about what genes were. We talked about transcription, which is making mRNA, and translation, which is making protein from the mRNA, and controlling gene expression in operons. We talked about conjugation, transduction, and transformation as far as gene transfer goes, horizontal, which is not from parents, not hereditary. And then we have different mutations that we talked about, as well as the Ames test. So that's it for chapter 9, and I will see you guys in chapter 10. I hope you'll be back.